Hi, um, I hope everyone is well. In the following hour or so, I will present the slides entitled Towards the Atomistic Model of RNP Core for SARS-CoV-2. I am Roden. I am a PhD student at Nalovis Group at KAUST, and the full PowerPoint is shared through this link. I have the page number at each page of the slide to help you locate the content in case you have questions or somewhere that I'm not clear or made a mistake. So my background was bioinformatics. I focused on DNA sequence analysis before, and I'm not a expert or experienced practitioner in structural biology field. So I might make some mistake. If that happens, please let me know. So this is the background. One part, the only background that I want to share with you is the RNA structure and its negative charge property. So this is the common visualization of the RNA structure that you receive from online or other resources like textbook. And these are the phosphate sugar backbone. These are the bases, AUCG. And if you go into atomistic detail, you will find phosphate at here, and it is negatively charged on this oxygen. And this RNA is built by plenty of these tiny building blocks, and so you will have plenty of those negative charges. And this is very important, as those charges will give the interaction forces within the molecule, so as to stabilize the structure. These are all the papers that I have read, and I will go quickly through each of them. I will only talk about the results of them. Will not focus on the methods or other like background or those information. So something to note is this guy publishes 4 papers on this direction, and this paper proposed the most detailed model, I think. And these last 3 papers are from bioRxiv. So the first one, it's actually coming from Scripps. And one thing to notice, the images in my following presentation have two mode. One is this one. They are shown in reversed contrast, which means the densities are in white, just like the text you see now. The first thing is the S and M and RNP complex shown here. And the relation between them is like this. And most of these N proteins, this RNP means RNA nucleocapsid protein. After the RNA bind to the N, we would call it RNP, and kind of form some complexes, we would call it RNP. This purple sphere here is one N protein, and another conclusion is from this paper is most N locates within 25 nanometer of the inner face of the membrane, which this is the spike, this is the membrane, and I would think here is the 25 nanometers of the proximal side of the inner inside the membrane. And this figure, I guess it's averaged from all of these figures and then did some statistical testing. There are some text here. So throughout my presentation, I will put some text nearby some figures, And there are some cases that I would not explain those texts, or I would not go very detailed into some figures. The purpose I put them here is either some of them I don't understand very much, or I haven't digged very further, but you might be able to read them by yourself, or if you find it very interesting and you want me to put more efforts on, please let me know. This one is the author observed a 15 nanometer strands. Like I cannot see it very clearly, but there are something different from the background at here and here, kind of like in this way, but not very apparent. And the author attribute them to the RNP. And this is a model proposed in this paper that those M proteins form some kind of lattices underneath the membrane, and this M protein interacts with the M protein, the C-terminal domain of the M protein, and then so that because it forms a lattice like structure at here close to the membrane, it's more ordered, and then there is also a disordered core further towards the center of the virion. And this is there are some numbers, like this is the 1s trimer, and then it will it will be together with 4 N proteins. This is another paper, the first paper on this topic that I found, published by this Chang Ke. The first thing is there are 5 domains for each N protein. This from N side to all the way to the C side is 1 N protein. This is another N protein. There are 5 domains, 1, 2, 3, 4, 5. There are 2 structured domains called N-terminal domain and C-terminal domain. And showing on the, this diagram shows the 2 N proteins form a dimer through NTD dimerization. And through CTD dimerization. Then shown on the right is the kind of a cartoon or a canonical representation of the structure details. So there are alpha helix, there are beta sheet, and those purple or blue shaded characters, amino acids, they are positively charged. You can see in this region, the N side, the very first portion of the CTD has a high density of those positively charged residues, which might indicate that they will bind to RNA, because RNA is negatively charged. So this is the C-terminal domain here. C-terminal domain structure, it forms a octamer, like observed by the authors. And these two images are the stereo pair of the octamer. And if I understand correctly, stereo pair means if you observe this image by your left eye, this one by your right eye, and then you somehow kind of you think it as something in 3D, and you might be able to reconstruct this 3D model. But I would think it might be too hard for the molecules for models in such detail, such as molecules. And then if you rotate this octamer by this direction by 90 degrees, you would see a X-shaped structure. and there are some dimension numbers. The width of this is 90 angstrom, which is 9 nanometers, and the height of this X is 7 nanometers. One thing to notice, the 8 monomers form 4 tetramers. I think there is a typo. It should be 4 dimers, 1 dimer, 2 dimer, 3, a 4th one. And it says 2 tetramers here. It's actually 1, 2 tetramers. I will show the model very quickly in PyMo. So this is the 2CJR PDB. And right now we are seeing this from this view. And if we rotate 90 degree, we need to rotate back. It's this one. So now we are seeing this view. You can see the relative position from within the 3D. There are some. These are some details of the structure that describe the forces, like either intramonomer forces within one N, intradimer, that's the dimer-dimer interaction. It's talking about those amino acids, the side chains that interacting with the other side chains. I will not go details into this. Like, as long as we have the PDB model, and then we can populate from the graphics pipeline. So I didn't put much efforts on this. Here is shown the surface potential of the CTD. So these two are from SARS-CoV CTD, but the length is different. This one is longer. It has on the N-terminal side of the CTD, it has more amino acids, around 30 amino acids more. And the author found that it will then form a positive groove at here. So they think this model is better than this one, and the people need to incorporate those amino acids in the N-terminal of the CTD so to do research. And this model is similar to another CTD, to the CTD of another virus called the IBV, which is a coronavirus as well. And also you can see that this binding pocket is slightly larger than this one. The author proposed a asymmetric helix formed by the CTD. In this figure, it formed by the 3 octamers. So 1 octamer, 2, 3, which means there are 24 CTDs in total. And it can represented by this 3x, but this x is different from the other 2. As you can see the color scheme here, this is the yellow subunit. It becomes to here. It goes to here in the second x. So if I understand it correctly, it looks like this. So if you put this one on a page, and then if you flip that page, just like you are reading it, if you flip that page and then you will get this. So just to show in PyMo is the reason you need to, like, the author would flip it, I think is because if you don't, you cannot get, there will be no interaction surface. So now we are seeing this one. if we just like pile this one at the bottom, you would find that this subunit cannot reach to this subunit to do the kind of interact with it. So that's why I think then the author kind of rotate in this way and then to form some interaction. That's my guess. We need to do some validation or ask the author the reason behind this. Then also you can see it is actually different from this surface potential representation. This and this and this, they are the same, but this and this one, it's not the same. Then from this helix, the author identified the author identified the pockets that can fit two RNA strands, and it's a left-handed helix. Like if you put your left fingers, follow the trend of the RNA molecule towards the up, and you will find it's a fit. If you go right hand, you need to like your fingers would not go up. The direction of your finger would not go up. So it's a left hand helix, so this RNA would bind the binding of the RNA here is the backbone of the RNA would lie at the groove, and then the bases will pointing out. And the author proposed, and also there are previous literature says that the NTD, so this is the CTD domain, the NTD has aromatic side chains that will bind to the bases to kind of, I coined it as protect those bases. So this structure is shown for this one. This is the sequence alignment of N termini of CTD. It shows the charge similarities. So first, the large number of positive charges shown in red, and they are highly conserved for those charge residues, and so the conserved region should have some functions, and the author proposed it binds to RNA. This is a paper on PNAS from 2008. It's the cryo-EM tomography for the MHV. It is not SARS-CoV. It's mouse hepatitis virus. It's also a coronavirus. First thing is they observed some circular structure at here, and it turns. So these are the different slices on the Z stack. So at slice 1, the circular is like this. On slide 2, it turns again, turns further. And they also observed some tubular fragments like this one and this one. So they proposed a model like this. These are spikes, M protein, tiny amount of E protein, and then this M protein interacts with this RNP. RNP forms some helix at here, and then like random structures at here. The RNP is interacting with the C-terminal domain of the M protein. The M protein, some of the papers call it a matrix protein, some of them call it a membrane protein. And the RNP is very flexible. It extensively twist and fold. There are some side notes from this paper. The first one is the 4 nanometer CTD-M layer after the lipid layer, which is this layer here. It is observed from the images, and this text describes why the author think it can attribute to the C-terminal domain of the M protein. This one describes the neighboring distance between M. It's 6.5 nanometers. Say this one, the distance, the space between these two M protein is 6.5 nanometers. I mean, not every pairs of M they are spacing 6.5, but it's a statistical observation. There are 3 movies from the cryo-EM tomography experiment. This one is for MHV, oh, sorry. I think I might make one mistake here. At least the 4th one might be not MHV, but what I want to show is they can form these movies. Oops, sorry. The 4th paper, again, by this author, it's on 2009. It is to investigate the multiple nucleic acid binding sites. Like there will be multiple binding sites found in this paper. Again, 5 domains, 3 IDRs. The definition of the IDR is here. The intrinsically disordered region, which means the CTD, NTD, they will have some structure, but the C-terminal domain, the middle part called LKR linker, and the N-terminal part, they will have no specific or fixed structure. And shown on the right is the experimental constructs used by the authors. We first look at this one and this one. These are the core parts of the NTD 45 to 181, and these are the core parts of the CTD 248 to 365. And the authors were going to flanking this core part by the C-terminal side or the linker side or the N-terminal side. They also have a full length, not full length, but a construct flanking CTD, linker, and NTD. So you can identify this by the numbers. These are some results from biochemistry experiment. The KD is the dissociation constant. The smaller, the stronger the interaction. And they are checking the interaction with RNA. So it's RNA binding affinity. They find the conclusion is that NTD and the CTD will bind better to RNA when either of this side or this side is present, and if the middle part, which is this part, the LKR, it is present, then it's the best, as shown here. And the bottom 2 figures shows the LKR region is a intrinsically disordered region, even after it binds to, even after within those constructs, it is linked to the CTD or NTD, it's still IDR. Then the author proposed a model. like after they did some SAXS experiment, the results is here. The model is like this, the CTD dimer in the middle. It's like a clamp. Just think as the CTD is my two shoulder, like two parts on the shoulder, forms a dimer, and then my arm would be the NTD, that kind of function like a clamp. This part is the linker, LKR in the middle, and the distance. The author stresses that we need to notice the difference between these two LKR lengths. Oh, one thing to say is the radius. I think if I understand it correctly, the radius is this one. The radius of the N is 61 angstrom, which is 6 nanometer. This is a description for the SAXS results. Another paper by this author and colleagues. The diagram of the 5 domain. The this paper it says the oligomerization of this helix is transient, and that oligomerization can still be able to form even they did the experiment by this construct that contains NTD, linker, and CTD. So in the previous paper, it only has CTD. Now even the author link the other two domains. It does not interfere. It does not affect this helix structure. I think, another thing is these colors here. You can see this blue sign, red and purple colors, these brown colors here. They are some mutations to mimic the function, one thing here, to mimic the phosphorylation on this LKR region. So you can see here, introduction of negative charges to the LKR through phosphorylation mimicking mutations in the dimer. I forgot the abbreviation for DD, but it means this didomain. So it has NTD and CTD, so it's didomain construct, and the oligomerization of that is enhanced. So there might be some regulation mechanisms by this phosphorylation at LKR. Please pay attention to this part. I will leverage this property in the model I propose in the final slide. One of the final slides. So the conclusion here is the oligomerization is transient, and it's driven by the neutralization of the charges, excessive charges on the protein, and it might be able to regulated by the environmental salt concentration and the protein phosphorylation. Another one from Chang Chong K. This is a review, so I will repeat some of the previous descriptions. This is a figure that dive from the virion structure to the cartoon representation that has S and M and N, and then further dive into the N details. It has an arm, NTD, linker, CTD, C tail, and the clamp structure at here. This is the sequence alignment of different coronaviruses, and they have similar structures. These arrows means beta strands. These cylinders are alpha helix. So those structures are highly conserved, and they also have conserved aromatic residues. If you remember from the previous slide, those aromatic residues are help to protect the RNA bases that point outwards to the helix. This is the sequence. This is the structural alignment of different coronaviruses. You can see they are overlapped. They are highly overlapped. So the structure is very similar to each other, and it also shows the positive charged region on the N-terminal domain. I forgot if I have talked about that before, but if not before, it will in the following some of the following papers. The structure of N protein. It has the another positive charge pocket, and the aromatic residues that help to protect the bases. As this is a review, the CTD positive charge region is shown at here. Also, the author compared the structure between SARS-CoV and IBV-MHV, other coronaviruses. A refined model of the helix RNP is first the bases are shown, it's pointing out, and then this NTD is shown. If you see it from side or the top, you can see where the NTD locates, and then the dimer-dimer interaction is weak, and this LKR is not shown here. The LKR linker would be accessible to interact with the matrix protein M, but there is no reference. I think this is just a kind of a hypothesis. Each N dimer would bind to 7 RNA bases. No references again. I think this is either the author play with it or another hypothesis. These two parts and also the weak property are very important, which I would talk later. This is a paper I found when I started to prepare my slides, so I didn't go very detailed. Here is the abstract. Basically, they present the 3D structures of a H coronavirus, another human coronavirus, but not SARS. The structure of N proteins, NTD, N-terminal domain. Previously, we were talking about the CTD, and that forms the helix. Now it's NTD. Complex with ribonucleotide, 5' monophosphate, which is the building blocks of RNA. And they identified a chemical called PJ34. It's an analog to the RNA building blocks. It can also bind to that groove, so it can serve as an inhibitor, which is related to drug development. So this is the structural overview of the binding between of NTD and AMP. AMP is the building block of RNA A, AUCG, those bases, nucleotide. This is A. It's shown at here, the surface potential diagram. These are some forces. You can see some atomistic detailed forces representation. And one thing very nice is we have the PDB model for each of the either A, C, G, or U binding to the NTD. It also has the analog, the inhibitors binding structure. Another paper, this is also I found after I started preparing my slides, but I found this one is actually is too good to be missed, so I included it. These slides I want to show a caveat that there are distortions in the negative staining images. So if you see on the left side is the negative staining images, if you can find some eclipse shaped virus, and I mean not all of them are a perfect sphere, but in the EM cryo-EM images, most of them are like they are better sphered shape. So we need to identify the images we used. If they are negative staining, there might be some artifacts. If they are EM, it's better. In this image, it's shown the RNPs and RNP fragments under EM. This is the RNP. I think by RNPs, the author want to say a larger structure or a larger structured or some characteristics we can see under microscope, and then by fragments it's a smaller one. So okay. So these figures, this H here is the negative staining. This one is J, it's cryo-EM. In both modes, we can see two groups of RNPs. The first one is the author coined as a compact intertwined flower shape. The other one is a loose filament shaped structure like this one. It's kind of filamentous. There is a strand, and the width of that is 15 nanometer. The same structure can be seen under the cryo-EM images. This is if they further examine by kind of some moving box, they move the boxes, a box through this strand, and they then do some average analysis. They find the RNP fragments show 7 nanometer apart strands formed lattice-like structure. So it's still 15 nanometer wide RNP fragments, and there are smaller strands, and the distance between each strand is 7 nanometer. The same thing observed under EM. This one is negative stain, yes. Then there involves some biochemistry, or the experimental details is these structures are observed right after the disruption of the virus. Then the images here is after the disruption, the author purify those RNPs and then put it in the refrigerator, and then for some time, I think it's 1 week or 2 weeks, and then image again under selective staining or EM. So these are the results after the, so they call it purified aged RNP. So these are the raw images and the averaged images. And then the author did a 3D reconstruction. These are reconstructed results. The surface mesh model. It shows the author coined it as a pot hook. Sorry, I didn't put it here. It's a pot hook model. Like it's hard to describe, but you can see from here. And then the author were docking the crystal structures of NTD and CTD to this pot hook model or mesh, and then they formed this structure. But there are some like some intensities or some spaces that is empty. Then the author added two distal ends to there, the C terminal end terminally and N terminal terminally. And this here is the author proposed positive charged group that binds to the RNA. There is one problem at here is the author only used NTD and CTD, but where is the LKR? So I think there's no LKR in this model. We still need to figure out the intact model of N. Then this is on the individual dimer level, and then the author also reconstructed the helix. So this is the helix proposed before by the Chang Zhongkei. And then if we feed this mesh, this pot hook structure here, then and then we add the N-terminal and C-terminal domain for each of the dimers, and then we will get this structure. And then if we pile it up, we will form this RNP helix. But there is one problem here. If you remember what I stressed before, is the direction of the middle X is flipped. It's different from other ones, from the neighboring ones. But from here, it is the same. Why it is the same? You can see there is a groove, red feature here, here, here. They are the same. They shouldn't be the same if the author follow exactly the previous model. So, and then the author gives the a RNA binding groove like this, and then now you can see this RNA binding becomes to a like a right hand RNA helix, is because this is going up, and then you cannot fit your left fingers. You can only fit your right fingers to this strand, and then it goes up. So this is inconsistent with the model before. Then the author proposed based on this model, the author proposed a supermolecule structure of the virus is first the dimer form the pot hook structure, and then it forms the lattices structure. So it forms the helix structure, and it will be seen under the microscope as lattices. And then it supercoils into flower shape, and then it is packaged in the virion. This is the cartoon representation of the model. This is now it comes to the SARS-CoV-2 model. SARS-CoV-2 research, they are all from bioRxiv. This paper is not open source. I put here only for education purposes. This is the sequence alignment diagram for SARS-CoV-2. You can see this SARS-CoV-2, SARS-CoV, and other coronaviruses. On the structural level, they are pretty much, they are very similar. And there are the representation of the alpha helix and beta sheet positions on the sequence level. This is the full length sequence alignment across different coronaviruses. The author found SARS-CoV-2 and SARS-CoV has almost 90% sequence identities. The author tried to examine the structure of the full-length N, but in one of the experimental steps, the N aggregates, which I don't quite get it. I didn't look into the details of this experiment, but it aggregates and it cannot perform the rest of the downstream experiments. So the author gave up the full-length N. And focus on the NTD of N. It's a right-hand model, but this is not a right-hand helix model. It's here. Why it's a right-hand? It's if you think this as fingers, these are basic fingers. The basic, if I understand correctly, it's there are basic amino acids there. And here is the palm, and then we have the acid wrist, and I think this might be the thumb of your right hand. This is the representation of the different secondary structure fragments, beta sheets, loop, and alpha helix. Where is? Oh, sorry. Within Oh, yes, there is one, eta 1. Yes, eta 1. Then this is the sequence alignment, structure alignment of different coronaviruses, NTD. It's very similar. This is the surface chart. No, this is the mesh model. I think it's. Oh, this is the structure alignment. It has the different color means the conservation level. The blue is the most conserved part. The red is the least conserved part. So you can see the blue, the blue are also positively charged, overlap with the positively charged region. So they might perform the RNA binding function. And these are the comparison of the surface charge between different coronaviruses. These are some detailed comparison between different models. And the author proposed that, and we can see from these diagrams that this binding pocket is larger in SARS-CoV-2. Like this steric hinder is less compared to others. These are some detailed interactions with AMP. To different SARS-CoV, so to different coronaviruses. And the author claimed that it revealed a unique drug target pocket, which I haven't read in very detail. The 10th paper, it's a very similar paper to the previous one, and it's published from some institute in Czech Republic. where I guess my advisor might have some like familiarity. This is the abstract. The four proteins, like the M, sometimes it's called matrix, sometimes it's called a membrane protein. And then this sentence is very interesting. It says the N-terminal NTD captures the RNA genome, while the CTD anchors the ribonucleotide complex to viral membrane via its interaction with the M protein. So here it says CTD interacts with M. But it has no reference. Again, this is the content of the paper. The CTD serves as a dimerization domain and binds to the matrix protein, but it has no reference. The SARS-CoV-2 model drawn in this paper, I think it's from this cryo-EM images. The author proposed this model, the spikes, the lipid, the M protein, this one is the nucleocapsid protein. The green ones are a single-stranded RNA. Yeah, the CTD domain of the M protein forms a dimer, basically the same as the SARS-CoV. Here is the solution structure of the SARS-CoV-2 NTD RNA binding domain. It's a right-hand model, like you can fit your right hand in this model. Basically, very similar to the one I described in the paper before. There are some, I think, biochemistry or some force analysis. In the end, the author proposed a model that the RNA duplex, so two RNA molecules, two RNA single-stranded molecules. I remember they are reverse complemental to each other. It's lies at here, and pay attention that the basis is pointing towards the N, and the backbone is at out. It is on the outer layer. So the backbone would lie at the helix groove of the formed by the CTD. If the previous model are correct. The final paper on bioRxiv on this topic is the RNA secondary structure. So the author identified 106 structured and 59 unstructured regions for RNA secondary structure. This is the location of the top 5, A, B, C, D, E, F, E, on the genome. A is at A is at here, here. B, C, D, E. Here it says the location of the structured and unstructured sequence or region on the genome. The sign is the structured loci, the yellow is the unstructured loci. There are some I think it's comparisons, the conservation analysis across different coronaviruses. There is one more paper I found when I prepared the slides, I was searching for the paper I read before, but somehow this one popped up. I haven't read it. The purpose of the paper is to find a drug to target the RNP, but and it also shows some structure details. Might be good to read afterwards. So far, like the overall picture is kind of formed in my mind, but there are some issues. The first issue is what forces is there that keeps the helix structure, this structure, like in this interface, what things are interacting with each other? I don't see a place that describes this, or I might have missed in some papers. And another possible answer is the RNA. Once the RNA wound or bind to this helix, it forms, because the RNA is intact, is a single strand, it forms some constraint. The second question is, which part of N interacts with M, CTD? After the membrane, there is a 4 nanometer layer that formed by M, and then it's the N-RNP complex. But what is the interaction there? Possible answers are CTD, that's in the bioRxiv paper, but without citation. The LKR in Chong case review paper, but no reference as well. And if we see from this structure, the NTD is at the outer side of the helix, so might be NTD as well. The third question, which is a very critical point, is how to compact such a huge genome. In the last paragraph of the 8th paper I presented, it has this statistical information that coronavirus, the diameter is 85, but most other reviews or papers I found is around at 100 nanometer. But the genome size is 30 kilobase. The HIV is much larger, and it has less it has shorter genome. HRSV and other virus, it's again much larger and smaller genome. I communicated with David for this, but here is the calculation from David, and I did the same calculation and got the same results. In short, we are calculating the densities and the number of N-dimers needed and the density of the volume of them, and we found it is impossible that so in Changcheng case review paper, it is said that N-dimer is there is one N-dimer every 7 nucleotide, but if this then if we assume that every nucleotide is bound to the N protein, then it is impossible, because that will need 4,000 N-dimers, and that this is the oops. It becomes possible. I've kind of forgot here. I hope I didn't make any mistake, but in copying from from my email to here. But the conclusion is the volume of the dimers is larger than the volume of the virus. And it's so from this comparison, from this table, we can see that it's very unlikely that they have the same genome packaging mechanism. So also, if I didn't make a mistake in copy and paste, then this variance volume is slightly larger than this one, but you still need to fit the RNA, give space for RNA, which is very unlikely that this can be fit in one variant. And also the average number, sorry, I didn't put it in my slide. The average number per variant, the average N number per variant is 1,000. Here there are 4,000 N dimers, which means there are 8,500 N protein, which is 8 times more than some statistical numbers in other papers. So after all of this, so what we should do is imagining from all of this information, the 25 nanometer within the membrane, the 15 nanometer filaments, the circular structure observed here, and it turns on different Z slices, the tubular shape, the RNA duplex interact with the NTD, the CTD helix, the CTD octamer, the NTD CTD clamp structure, the this is followed by this overall model proposed by one of the authors, this model proposed by another author, this pot hook structure for NTD, CTD, NTD, this supercoil, this flower-shaped structure, and this RNA secondary structure. But these are very few. There are only hundreds of them, and which has like at most thousands of RNAs, which is less than 1%. I think less than 1% of the genome. So we need to imagine a model that can conform most of these observations for RNP core. These are some of the attempts that I tried to like these days. The first one is inspired by these numbers. The most N are located within 25 nanometer of the membrane proximal side. And another, this calculation, 7 times 3, 7 is at here. Each octamer, the height of the octamer is 7. So 3 of them would be 21, and plus 4. 4 is the, if you remember, it's the distance, the space of the layer after the membrane. There is a it's comprised of CTD domain of the M protein. So if we add this together, it's 25. So this is the very first model that's in my mind. It's this structure is interacting with M right after the membrane. But the problem is, if it's a tight, compact model here, we should be able to see some ordered structure in the EM images. Even if they are like this, they are spaced apart from each other, we should still be able to see it, just as we saw the M protein in the membrane in the same paper as where this figure from. Another thing, another contradiction here is if it is formed in this way, if it is oriented in this way, then we wouldn't be able to see the turns, the turning circular structure at here, because most of the things we saw should be in this direction. We should saw this strands, these tubular strands near the membrane. So this is unlikely. This is another model that I thought that came into my mind is inspired by the structure, by the tetramer structure in one of the entities published recently, 6VYO. I don't know why the author put 4 subunits that forms a tetramer here, but this figure inspired this model. That so this is NTD, this is NTD, so the NTD interacts with each other, form kind of a tetramer here, and most of them like they are tightly compacted here. This has the similar problem. Is we should be able to see some of the structures under cryo-EM. So then this is really out of imagination. It might not be true, but it's one of the attempts in my mind. So I coined it as a pre-nucleosome non-uniform beads on string parking model. I will explain step by step. So on the left side of the slide is the from textbook. It's the nucleosome, it's the DNA packaging hierarchical flow. So this is the DNA, this is the nucleosome formed by histones. I remember 8 histones. It's called a beads on string. The distance is there are some statistical numbers. I remember it's 147 nanometers. Sorry, nucleotide. Or around 150 nucleotides. So it's a fixed length. Sorry, so it's not a it's not random dotted beads. It's a fixed length beads on string, and then it forms this 30 nanometer chromatin fiber, and then even higher structure and condense and form chromosomes. I remember from some lunch, from some conversations on the lunch table that this part is not clear, and this part is being challenged. Some zigzag model that being challenged, but the thing is, it's uniformly spaced nucleosome beads, and it is very tightly compacted. There are no DNA that's protruding out. Not much. So inspired by this, I think this one, I call it pre-nucleosome, is because it's non-uniform. It's not uniform distance spaced nucleosome. So this, I use this one to represent a dimer. This blue string represents the RNA. So if we line it up on a straight line, we will see a dimer, another dimer, third one. The distance between them may not be the same. Sorry that I put it like they are the same. And a tetramer, oh, sorry. This is a former of the dimer. So it's actually an octamer. So this is tetramer of the CTD N dimer, and all of them, they can slide. I call it, they are driving on the RNA. Once they drive and meet another octamer or tetramer of the dimer, they collide with each other, they stop, or three of them driving together and they form a longer one. There are some dissociation or association constants, like between them, the interactions between them are weak, so they might fall or they might drive separately in different directions. If like I simplify this diagram to this one. This represents this dimer. This one represents this octamer, and this like I use this rectangle to represent the helix structure. This I want to say these are the helix structure from like here or here. And this should include, okay, so this one should be better characterized by this, but the direction should be changed if we follow exactly this model. Then they are within the virion, and then I have a term called homeostatic parking. It's because the C-terminal domain can interact with the RNP. So the RNP would try to park. They are driving within the virion. They will try to park on the membrane. So it's a moving thing. It's not static. And the park and the dissociation, it's it form some kind of equilibrium and becomes a homeostasis like process. And the one distinct thing here is there are free RNAs. Not every RNA are bound to this RNP. Like so there are single dimers, there are octamers, and interns at those turning points. There are supercoils as well, and there are secondary structures somewhere. The spatial ratio is not true here. This secondary structure should be very small. So by this, then we can see there are some. So this will follow or conform to the observations before. Most observations before, it's there are tubular structures, there are circular structures here, there are secondary structures, like all those things. But this is inside virion. How to explain the structures observed here? This is after the virus is already disrupted into some solutions, some experimental solutions, or some, like it has some salt concentration or some other things. Like this is different from this. Like we cannot see a clear flower shape within the virion. Why is that? So if you recall that there are some phosphorylation site on the LKR between the, this is LKR, there are some phosphorylation site here. Those phosphorylation can regulate the dimerization, the formation of the helix, the RNP helix. So and those salt concentration will change it as well. So my hypothesis is that after it disrupts, it quickly, like the environment changes, either the salt concentration or there are some phosphorylations on the LKR or some other reasons. The interactions between the dimer, those weak interactions became stronger. So it forms a supercoil, a much tighter supercoil that gives us the flower shape in the in one of the papers. And in this case, so this structure, this flower shape might be also in the similar in the cell in the ER Golgi intermediate compartment. That's where the budding happens. If I am correct, I only read one paragraph about that. There, it also might be this structure at there, but the thing is, it loses N when the virus buds. So inside the cell, after the replication of the genome, like all the RNA is protected by the RNP. Then when it tries to bud from the cell, the N will lose because of the volume constraint. And then how to explain the virus that invaded to a cell? Right after the invasion, there will be some of the RNA. It's naked. It's exposed in the cell that will be cut by the immune systems inside the cell. So my hypothesis, like how to back up this is I think it's okay that it's being cut, because there are still some other open reading frames that can function. And another thing is you have multiple variants. So the variant can only do some harm to you once it reaches some concentration, because you have more variants, and then you will have a chance that some of them are intact and finish the replication and then bud. So this is not a perfect model, but it's one of my hypotheses, and can explain some of the observations before. There are some to-dos. The first one is build the helix model, or we ask the author, which is this model. Which is this model here, or this model. And we need to fix that if we conform to this one. I have done some structural prediction of the by put the full length N into Swiss model, and it gives me two separated region. I think one is NTD, the other is CTD. So right now we have the structure of the NTD, either the NMR structure or the crystal structure for the NTD for SARS-CoV-2, and not the CTD. I'm not quite into this like this structural prediction thing, this different parameters here. I would hope I can get some help at this part. Then the second task is to wind the RNA on the helix at here, and then to see how many RNAs we can feed per 24, or say per 24 those RNP helix structure. And then my personal feeling is the NTD at this layer here, this NTD will interact with MCTD because it's in the outermost layer or the LKR. So we might be able to do, but the problem of that is the problem of LKR interact with CTD domain is LKR has no specific, it's a IDR, it's an intrinsically disordered protein, so it has no interacting domain to the C terminal domain. Might have no, but might have as well. So I think we can do some prediction, the protein-protein interaction prediction by NTD and MCTD, or LKR and CTD as well. There are some implications of this model, like we can find inhibitors to break the dimer interactions, or the or to break the octamer interactions, or to break the CTD with interaction with the RNP. Another one is we can break the regulation, like the switch here. We can break the switch, like the phosphorylation should be achieved by some kinase, I guess. If we can block that kinase, we can block this RNA genome package. So that's all. Thank you very much for listening. All kinds of feedbacks, like questions, comments, suggestions, or discussions are very welcome. Please send it to my email, and stay safe. Thank you. Bye.